It gives me great pleasure to introduce our next speaker, Peter Hahn. Peter is the Israeli representative of the Beijing Genomics Institute. The Beijing Genomics Institute was founded in 1999 with the vision of using genomics to benefit mankind and has since become the largest genomic organization in the world, with a focus on research and applications in the healthcare, agricultural, conservation, and environmental fields. BGI has an improving track record of innovative, high-profile research, which has generated over 1,847 publications in many top-tier journals such as Nature and Science. The title of Peter's talk for us today is The Application of NGS on Life Science Research. Peter, when you're ready.
It's great happy for me to see you here, and maybe some of you are already familiar with me. But today I want to introduce something about BGI and BGI tech solutions for biology research. BGI formerly is called Beijing Genomics Institute, but now we just call BGI Shenzhen or BGI Hong Kong, BGI somewhere. And it is founded in the year of 1999 on the September 9th. Actually, now BGI is only 17 years old, but it is already the biggest genomics institute in the world. At the beginning, BGI was founded with the mission of China in the Human Genome Project. We just took one personal responsibility in that project. After that, we do 10% in another project and 1% in our own project. Now, BGI has dozens of branches in the world. We have a laboratory in Denmark, in Japan, American, and now we have 47 laboratories in the world, and about a half of them are working on clinical service. BGI have 9 branches, and this is we call it NGS for the clinical service. This is for tech solution service for you. Yeah. And also we have our BGI agriculture. When we have a good project, have a good cultivar. Actually, for BGI agriculture, we are now commercialize the millet and group. I will show you some picture about this. And for BGI research, this is the BGI main direction. And also, we have acquired a company in the year of 2013. Now we have our own sequencer for research and clinical service. Here is about data science. It's open. It's free access journal for all of the scientists in the world to share their new research, share their data. This is about the staff of BGI. It's young, active, and but still very, very expensive, skilled and experienced. We have published more than 1,000 papers on SCI journals, and more than 260 papers on CNS, Cell, Nature, and Science. Yeah, from Human Genome Project, we developed a step-by-step from 1% to 10% to 100%. And after that, we find that our power is not so strong to handle a very big project. So we initiated the Human 1000 Genome Project with the scientists from UK and American. And now there are 6 countries, there are scientists from 6 countries are working in this project. This is also the biggest human genome data in the world for service for all of the research for human genome, human genetic research. Yeah. Now this project has published more than 3,000 more than 3,000 individuals genome. And after that, we have other projects such as the Single Danish. It's our collaboration with the Denmark team working on the metabolic disorders. And also, we are a member of International Cancer Genome Institute. Here I want to emphasize some of them. This is about our collaboration with eTwin. In this project, we want to sequence 5,000 couple identical twins to find what is the difference on their epigenetics level, which result in the different phenotype when they are growing up. And for this project, we want to sequence 3,000 trials to find what is the de novo mutation or what is the novel reason cause the autism in the child. This is about our collaboration with the International Rice Research Institute. We have published more than 3,000 types of rice. Actually, the samples are from the from Egypt, and they have a bank for all of the wild and cultivar rice traits. So we almost thoroughly sequence all of the rice types in the world. We have published this data for free to all of the research community. At the beginning, we worked on a space or a new family, but now we sequence more than 40, I forgot the exact number. If I'm not wrong, it is 47 words to do something thoroughly in a specific family. Yeah, we published the rice genome database. If you are interested or if you have any problem to access this database, you can contact me. Now we are working with more than 15,000 researchers from more than 2,000 institutes or companies or universities in the world. And in Israel, we are working intensively with Tel Aviv University and Hebrew University. Also, they are all. Now, all of top 20 pharmaceutical companies are working with Beijing. We have almost the full range of full levels of technology, technology platforms, from DNA sequencing to cell clone to cell platform or even cloning platform. This is about our facilities. We are doing something. You are researchers. You do your experiments in your laboratory. We are doing. We just move the experiment to the factory. Yeah. To do something. To do research, like industry. And we have the most comprehensive QC steps. This is a demo for whole human exome sequencing. This is about our certifications. We have a CAP certificate in Hong Kong laboratory. Also, we are authorized user from Illumina, from engineering, Beijing, and other suppliers. This is about our ISO. Yeah, we have successfully held the conference on genomics. This is we call ICG. This year is 11th. Yeah, this is about our new machine. This is after we acquired the complete genomics, we have to do some improvement, and now we this is the latest version of BGI. Now we already have developed the application on this platform for Nifty, for exome, for RNA. In the future, we will develop more applications on this platform. Yes. On September 9 is the birthday of BGI. We have finished almost 2 million samples on HPV, 1.5 million samples on Nifty, and also other tests we have do a lot of samples. Actually, for BGI now it's harvest season. We have our own machine. Our millet is ready, and this is about our hybrid group. For both our millet and group, we are doing hybridization, not the transgene or other technology. This is the China National Gene Bank is operated by BGI. Yeah. We always have dream. At the beginning, we have dream to finish the human genome project to take only one person. Now we have many directions. We want to work with you to have a better life for the human in the future. This is about BGI. And to introduce BGI is not my object today. Now I want to introduce you about applications of BGI. That is for which technology or which service we can provide to you. Okay, it is for the life cycle of the DNA record. Have the genetic information and translate into RNA and protein. So from DNA level, we have service on different platforms from microarray, from mass spectrum, from sequencing. And we are doing genotyping, some very general and very easy service. And also we are doing very difficult ones, something like whole genome sequencing, de novo genome sequencing for new species. Yeah. For this project, it may cost you hundreds of thousands of dollars in the past days, but now we are doing at a very competitive, very reasonable price with new technologies. And also we provide very common services for the transcriptome analysis, such as de novo transcriptome and whole genome, whole transcriptome sequencing, or just to computation. Some of you are a little confused by, I just need RNA-seq sequencing, RNA-seq service. Why you tell me transcriptome sequencing or computation? What is the difference between them? If you are interested in these topics, we may discuss. Actually, for example, if you are interested in the expression level, only interested in the expression level of transcriptors, we can do. If we just focusing on the expression level, we don't need to sequence the whole transcript. We just use a tag and just count the number of tags for each transcript, and use the number of tags to further analytic bioinformatics to recover the different expression level for genes, for transcripts. And for epigenomics or epigenetics service, we have whole genome bisulfite sequencing, we have RBS or medium sequencing, we have chip sequencing, and now we also have the targeted bisulfite sequencing. It means if you are interested in a specific region, you want to know the methylation conditions, methylation level, or something like this. In a specific region, we can do targeted methylation and to do sequencing and analysis for you. Yeah. This is a new technology from us. And also for proteomics, we have generally 3 divisions. Sorry, another one is not clear here. We can do proteomics profiling to know the general outline or full picture of a sample. And after that, you may interested in the level of competitive comparison level between different samples, so we can do quantification on specific list of proteins. And after that, we can do targeted analysis on specific proteins or specific peptides. For DNA sequencing, we have some of them. Yeah. Actually, today I only have 40 minutes, so I cannot explain every details with you. Actually, even for each topic, I can maybe introduce you for 1 hour or 2 hours to explain. Besides this, we can do non-cloning RNA. It's a new service from Syntezza. And this is about some epigenetic service from Syntezza. Actually, small RNA is a part of epigenetics. but we divided it into RNA sequencing. Yeah, formally I introduced you so much about DNA sequencing. Today I want to emphasize to spend more time on the proteomic service in design. Generally, we have several derivations. Yeah, this is the profiling. It gives you the result of the general global information about a specific tissue or specific condition. This is about the quantitative analysis on a specific list of proteins. Yeah, this is a pipeline. So we can do it in laboratory or lab. Can do add samples together. This is about the targeted proteomics on specific peptide or protein. We also do very general mRNA. Yeah, actually, BGI not only provide a specific service on a very specific topic. Also, we can provide integrated solution. When you find a gene, you may want to know the expression. A mutated gene, you may want to know the expression of this gene. So we can do RNA sequencing and even to know the modification of this transcript. This is the focus of BGI service. We want to provide you service at a reasonable price, good turnaround time, and always in high quality. The BGI project may be a little later, maybe not at a very competitive price, but BGI service is always in high quality. No client complain with me about the quality of BGI. They may say, Peter, your price is higher than others, or why it's so later, you didn't give me the result, and they never complain the quality from me. Yeah, actually, you can see this because I don't have so much time. Also, it's a limitation of my knowledge of our service, or because I'm not a real researcher. Yeah. So if you are interested in BGI or interested in the collaboration or cooperation of our service, we can discuss later. Yeah, thank you. Only a service provider, we can also partner with you on pure scientific research. Because I have introduced BGI, we have a department on pure research. They don't earn money. The object is not to earn money. They just do research. So if you are interested in any direction you want to work with BGI, you can contact me, and I introduce our scientists to discuss with you.
Thank you for your lecture. I want to ask the officers you showed in the map in Europe, that collaborate with BGI, or it's functions?
Your office
There was a map when we were talking about the collaborations and the projects, and you showed there was Copenhagen and Amsterdam. Is an office in Europe?
Actually, we had office. We even have a sub headquarters in Copenhagen.
Okay.
Just because the founder of BGI is graduated. He's graduated from Copenhagen University.
Ah.
So we have some researchers are still working in Copenhagen.